Everyone, it's 11 o'clock here in Bloomington, Minnesota, so we're going to go ahead and get started. Thanks everyone for joining the webinar today. My name is Jackie Carville, the Marketing Director here at ImmunoChemistry Technologies, and I will be moderating this webinar. You may have noticed that your phone has been muted. However, we do encourage you to ask questions along the way. Simply type your question into the chat dialog at the right side of the screen and select Send to Host. If you have any technical difficulties during the webinar, please send your chat to me and I can do my best to get you up and running. We will be posting the video recording of this webinar later this afternoon or tomorrow morning, so you can go ahead and look out for an email from me after the webinar with a link to that recording. Now I'd like to go ahead and introduce our speaker, Sally Head. Sally is the president here at ICT, where she provides strategic direction and oversees the day-to-day operations of the company. Today, she's going to be going over some flow cytometry-based assays to understand cytotoxicity. And with that, I'm going to go ahead and turn it over to Sally.
Great. Well, thanks, Jackie, for that nice introduction. And hello to everybody out there, and thanks for joining us today. I'm Sally Head, and I'm the president of ImmunoChemistry Technologies. I have a degree in genetics and cell biology from the University of Minnesota and an MBA from Cardinal Stritch University. I started at ImmunoChemistry in 1996 as a lab assistant and have been involved in almost every aspect of the company since then, including the development of our Cytotoxicity Kits and apoptosis detection reagents. It's hard to believe I've been at ICT for 20 years. It's a long time, but it really doesn't feel like it. You know, there's always something new and interesting happening with our products and how our customers use them. ICT has been around since 1994, so I got involved with the company when it was still a startup. Now I'm an owner, and since most of our coworkers are women, ICT is a woman-owned and operated small business in science. We offer contract services and sell some research products. Our lab services include immunoassay development and manufacturing and related services like custom conjugations and lyophilizations, plate coating, etc. And as a branch of our assay services, we sell ELISA buffers for researchers who are making their own assays, like coat buffer, block buffers, conjugate stabilizers. So if you need an assay, give us a call. We can help you make your assay better. And because we love cell biology, we develop several assays for whole cell analysis. These kits are typically not ELISAs. Instead, they use cell-permeant fluorescent reagents to analyze whole, living, intact cells. These assays detect apoptosis and cell death, including cytotoxicity. So today I'm going to talk about how to quantify cell-mediated cytotoxicity using flow cytometry. We'll review the immune system a bit and talk about some of the traditional techniques and include ICT's cytotoxicity and apoptosis reagents and how they can be used to quantify the cell-mediated response in whole cells using flow cytometry to help understand the effects of drug treatments and viruses. And then we'll take a few questions. So let's begin. Cytotoxicity is something that kills cells. It is toxicity caused by chemotherapy or other agents or actions harmful to the cell that kills it. Cell-mediated cytotoxicity occurs when cells of the immune system, like the effector cells, act on another cell, which is the target cell, to kill the target cell. In this picture, two natural killer cells, which are the yellow ones on each end, are attacking a red cancer cell in the middle. The NK cells, the natural killer cells, are effector cells. They are having some sort of effect on the other cell, and the red cancer cell is the target cell. That's a cell that's reacting. In this case, it will eventually die. This is the kind of cell-mediated interaction that is of interest to us. You know, are those NK cells killing the tumor? And in this case, yes, they are. These are actually genetically engineered cancer-killing NK cells from the University of Minnesota, which someday could become an off-the-shelf anti-cancer therapy. So we're doing important stuff. Well, how does the cell-mediated immune response work as part of the immune system? The immune system keeps you healthy by replacing old cells, checking for damaged and cancerous cells, and by detecting foreign invaders like viruses. The first arm of defense is simply a physical barrier. You know, the wall is your skin and mucous membranes that protect the body, and it can also be a chemical barrier like lysozymes. And then there are the innate and adaptive immune responses. Cytolytic activity is an important process for eliminating intracellular pathogens in cancer cells and has both like the innate and adaptive effector cells. So using the pathways of the innate immune system, the body launches an immediate and nonspecific attack with cell-mediated and humoral components. Cells involved include natural killer cells, mast cells, eosinophils, to name a few. And there is no immunological memory. On the other hand, the adaptive immune system takes a little bit longer to respond, sometimes like 5 days, but generates a specific response which is memorized by the immune cells, which is why vaccines work. This involves B and T lymphocytes, which include the cytotoxic T lymphocytes, CTL, which I'll mention quite a bit today. And like the innate system, has both cell-mediated and humoral components. Natural killer cells and cytotoxic T lymphocytes are the central mediators of cell-mediated cytotoxicity and play important roles in the protection against intracellular pathogens and cancerous tumor cells. As part of the innate immune system, the natural killer cells is a type of cytotoxic lymphocyte. NK cells in the innate system, which again generally is a fast and non-specific response, play a role that is analogous to the CTL cell, which remember is part of the adaptive immune system. It takes longer, and I'll discuss that next. So looking at the cells in the middle of this graphic, normal target cells are protected from killing by NK cells when signals from stimulatory ligands are bounced by inhibitory signals from the self-MHC class I molecules. If a target cell loses expression of the self-MHC class I molecules, as in the left cells, such as a result of transformation or cancer or infection or a virus, then the stimulatory signals from the target cell are unbalanced, which activates natural killer cells to kill them and results in lysis of the target cell. This is known as missing self-recognition, which I tend to think of as guilty until proven innocent. The NK cells will kill a target cell unless the target cell has the self-MHC class I to prove it's okay. So looking at the cells on the right, transformation or infection or virus might also induce expression of stimulatory ligands such that constitutive inhibition from normal inhibitory receptors is overcome, and that is known as induced self-recognition. So in many cases, it is probable that both the missing self-recognition on the left and the induced self-recognition on the right operate simultaneously to provide NK cells with the maximal ability to discriminate between normal cells and transform or infected target cells. So NK cells kill the target cells by lysis or apoptosis, and as you can see the little dots in this picture. The NK cells contain small granules full of pore-forming molecules such as perforin and proteases like granzymes within the cytoplasm. So when in close proximity to a cell that it wants to kill, the NK cell releases these granules. The porphyrins form pores in the target cell membrane, which creates a channel for the granzymes to enter. The granzymes and other cytotoxic molecules go on to trigger either apoptosis or osmotic cell lysis, and this distinction is really important. So in dealing with a virally infected cell, the last thing you want to happen is for the infected cell to lyse and release all of the virions contained inside it and infect the rest of the body. Apoptosis would be a better option, resulting in the death of the cell and the destruction of the virus without releasing any of the virus inside. It's a controlled process, and unfortunately, some viruses have adapted ways to circumvent the apoptotic process, thus impairing the function of NK cell and spread, which I'll discuss in a minute. So back to CTLs. As part of the adaptive immune pathway, which again takes longer, but it has immunological memory, the cytotoxic T cell is a type of T lymphocyte that kills cancer cells or intracellularly infected cells, which is typically caused by a virus. And cytotoxic T cells have many different names, such as cytotoxic T lymphocytes, the CTL, the T killer cell, the cytolytic T cell, CD8 positive T cells, or killer T cells, so you might hear about them quite a bit. Most cytotoxic T cells express T cell receptors that recognize a specific foreign antigen that may appear on the target cell. An antigen is a molecule capable of stimulating an immune response and is often produced by cancer cells or viruses. And once inside the cell, the antigens are bound up to the MHC class I molecules and brought to the surface of the cell, where they can be recognized by the T cell. If the T cell receptor is specific for that antigen, it will bind to the antigen and the class I MHC complex, causing the T cell to destroy the target cell. This is similar to the NK cells of the innate pathway, and again, here you see little dots. The CTLs release perforins, which form pores for the granzymes and the granulysin to enter the target cell and subsequently trigger apoptosis to dismantle the target cell in a controlled manner without spreading the infection. Oh, well, that was the slide I was talking about with the CTLs. Looking at apoptosis, so again, when cytotoxic effector cells like NKs or CTLs recognize a foreign, cancerous, viral, or bacterial infected cell, they'll kill it by lysis or apoptosis. So apoptosis is an active programmed process of cell death which dismantles the cells from within and avoids eliciting inflammation. Apoptosis is typically initiated through two pathways. There's the intrinsic pathway and the extrinsic pathway. The intrinsic pathway is activated following cellular stress, resulting in mitochondrial perturbation and the release of cytochrome C. Once released, the cytochrome C binds to the apoptotic protease activating factor 1, which is APF1, triggering the formation of the apoptosome complex, resulting in the activation of initiator caspase 9, which then activates the effector caspases 3, 6, and 7. The extrinsic pathway is initiated following the activation of death receptors of the TNF superfamily, and the trimerized receptor ligand complex recruits procaspase 8 instead of caspase 9 to the death-induced signaling complex, also known as the DISC, via the FAS-associated death domain, the FADD, leading to the rapid activation of initiator caspase 8 again, which then activates the same effector caspases 3, 6, and 7, which are responsible for the phenotypic changes associated with apoptosis. So during cell-mediated cytotoxicity, the cytotoxic T cell releases granzymes, which enter the cytoplasm of the target cell through the pores made by porphyrin. The granzymes, which are serine proteases, can directly trigger the caspase activation cascade leading to apoptosis, and alternatively, apoptosis can be induced through the cell surface interactions between the cytotoxic T cell and an infected cell. When a cytotoxic T cell is activated, it starts to express the surface protein Fas ligand, which can bind to Fas molecules expressed on the target cell. Engagements of Fas with Fas ligand allows for the recruitment of the death-induced signaling complex and the Fas-associated death domain. It translocates with the DISC, allowing recruitment of, again, procaspases 8 and also procaspase 10, which then activate effector caspases 3, 6, and 7, which again are the same, leading to the cleavage of the death substrates. The final result is apoptosis of the cell that expressed Fas. So here's a complex slide of some of the viral products that circumvents the immune response. Since viruses are potent activators of apoptosis, these organisms have evolved a variety of strategies to prevent these events. Normally, NK activity can be stimulated up to 100 times by interferons, but viruses have developed elaborate strategies to inhibit IFN antiviral function and the mechanisms of cell death. So mice lacking functional IFN systems are extremely sensitive to lethal infection with a number of viruses, including vesicular stomatitis virus, which is VSV, the Sem leaky forest virus, and vaccinia virus infection. A number of virus-associated malignancies and hepatitis C virus infection will respond well to IFN treatment in experiments. However, IFN is not really an ideal therapy as it has other negative side effects. So many viruses can also inhibit MHC class I expression and host protein synthesis, thereby preventing the natural killer cells from distinguishing infected from non-infected cells. And the role of NF-kappa beta is also important in controlling cell death along with P53. NF-kappa beta may be regulated by a number of viruses such as Epstein-Barr, resulting in enhanced survival of the infected cell. Several BCL2-like proteins are encoded by many viruses, again, such as Epstein-Barr and human herpes virus, to inhibit mitochondria triggered by cell death. For example, herpes viruses and cowpox viruses and insect viruses encode products to inhibit efficient activation, while gamma herpes viruses inhibit mitochondrial input into the apoptotic process. The DNA, such as herpes, also circumvent this immune response. So this process of apoptosis and lysis through cell-mediated cytotoxicity occurs constantly throughout the body under a wide range of circumstances. It's generally involved in host defense mechanisms, response to drugs or chemicals or viruses, like I just discussed, or other biological factors like CTLs targeting virus-infected cells or targeting melanoma cells, HIV infection, diabetes, and tuberculosis research. Immunotherapy drugs and antiviral treatments often work by stimulating the body's immune system, and a better understanding of the mechanisms of cell-mediated killing will help improve these treatments. A couple of things to keep in mind when testing for cytotoxicity. So remember that cell-mediated killing through innate and adaptive immune responses involves triggering cell lysis as well as apoptosis. If using an assay for just necrosis, it will miss those cells in early apoptosis and thus underestimate the amount of cell death occurring. Like in this example of natural killer cells, you can mix the cells together and see how they act upon each other, or you could just add the drug to the target cell population and measure apoptosis and necrosis. So you want to know, is the drug, virus, or other treatment influencing the response? Does that treatment stimulate or inhibit the response? How do you measure it, and how does that correlate with cell viability or cell death or apoptosis? Does the test generate cumulative data, as in a plate well, or does the assay look at individual cells? What's the timing of the assay? Does it cross-react with something else? Basically, are you getting the answer that you want? Instruments typically used when testing for cytotoxicity are microscopes, plate readers, flow cytometers, scintillation counters. Most labs will have access to this equipment. Well, here are some assays that have been used to determine cytotoxicity. The top four are basic assays that will generate simple live dead data as general indicators of cellular metabolism. They typically measure the release or reaction of an enzyme or metabolite. However, these reagents may be leaky and often require cells to be lysed. These assays can work well when labeling all cells in the sample to generate cumulative data, like in a plate reader. However, they can't distinguish between target and effector cell death and which cell type is actually releasing that metabolite. They can't distinguish between dead cells and those in the process of dying. And one of the big limitations is that they underestimate the amount of cell death by excluding apoptotic cells. ICT's cytotoxicity kits allow for the easy quantification of cytolytic killer activity without the need for lysing the cells, waiting for this enzyme release, or using any toxic radioisotopes. They can easily differentiate target cells from effector cells when grown in cultures and enable researchers to measure cell death at the single cell level using flow cytometry to quantify the four populations, live, early apoptotic, late apoptotic, and necrotic target cells, which I'll explain a little bit more later. Now, one of the oldest methods for detecting cytotoxicity is using the radioactive chromium-51 release assay, which was developed in 1968, a while ago, and is still used today. Target cells will passively internalize and bind chromium-51 from sodium chromate. The effector cells are added and will lyse the target cells, thereby releasing that radioactive probe into the cell culture supernatant. The amount of chromium-51 bound inside the cell compared with the amount of free chromium-51 in the supernatant indicates the number of lysed target cells. While this assay can provide useful quantitative data, there are some major drawbacks regarding the lab safety, the high cost of running the assays, along with the detrimental effect of radioactivity on the cell function, which may skew results. The chromium-51 release assay is also susceptible to wide variations in radioactive labeling and exhibits a relatively high level of spontaneous chromium-51 release. As a result of these drawbacks to chromium-51, other release assays were developed which utilized non-radioactive components such as MTT, endogenous lactate dehydrogenase, and ATP. For example, the MTT assay is a simple colorimetric assay analyzed in a plate reader. Yellow MTT is reduced by active dehydrogenases in the mitochondria into the insoluble formazan product, which is purple. So if the cell is living and has functional mitochondria, MTT will turn purple. Next, the cells are lysed and the signal is read in a plate reader. This technique can determine the loss of viable cells and a reduction in proliferation. However, in some cases, this increase in proliferation may be offset by the amount of cell death. So you can't tell if anything's happened. So these types of release assays suffer from high spontaneous release of the tracker diet from both the target cells and sometimes the effector cells. They may generate false positives or high background. if the target and effector cells release the same component like LDH. And these assays can't identify target cells nor distinguish apoptosis from necrosis. Flow cytometry assays have been developed to overcome some of those negative aspects associated with the simpler release assays. Flow cytometers are quite common, yet very sophisticated, as they can count individual cells and multiplex with several reagents at one time. Flow assays usually consist of a fluorescent membrane stain to label the target cells and thus differentiate it from the effector cell population, as well as a fluorescent vital stain to detect the dead and dying target cell population. Flow assays of this design have several advantages over that simpler release assays because they eliminate the use of hazardous radioactivity. There are no detrimental effects due to radiation on the effector cell population. There's shorter incubation times, some lower cost. You can differentiate subpopulations of cells. You can analyze single cells, and you can assess the viability of the effector cell population as well as the target cell. Building upon these older techniques, ICT has improved the flow cytometry assays by combining a membrane stain with a necrosis stain for our basic cytotoxicity kit, and then we added an apoptosis reagent to create the total cytotoxicity and apoptosis kit. ICT's kits allow for easy quantitation of cytolytic killer activity without the need for lysing cells or waiting for enzyme release or using radioisotopes. They easily differentiate target cells from effector cells when grown together in cultures and enable researchers to measure cell death at the single cell level using flow cytometry. The total cytotoxicity kit offers significant advantages as it can concurrently measure necrosis and apoptosis at the same time to quantify the four populations of target cells. Again, the live cells, early apoptotic, late apoptotic, and necrotic target cells. ICT's basic kit distinguishes live target cells from necrotic target cells without interference from effector cells. It measures necrotic and membrane-compromised cells in a mixed population sample of effector and target cells when grown together. It contains an assay buffer, CFSC, which is a green membrane stain, and 7AD, which is a red live dead stain. The total cytotoxicity and apoptosis kit includes all the components of the basic kit, plus it includes our SR flicker reagent to label apoptotic cells, kind of an orangey red, leading to a more accurate assessment of cytolytic activity. To use the basic kit, first you would label the target cells with CFSC, which is a green fluorescing membrane stain. If you're studying the effects of a drug or toxic agent, this CFSC staining can be optional if you're not actually mixing them together with the effector cells. Next, you would add the unstained effector cells, such as NK cells, CTLs, CD8 positives, or PBMCs, and incubate with the target cells. This mixture is referred to as the effector to target cells, or ET mixture, and you want to add those together at certain ratios. During the incubation period, the drug or effector cells will act on the target cells, and after this incubation, add 7-aminoactinomycin D, which is 7-AAD, which is a red fluorescing live dead stain. This will enter the cells with compromised membranes and bind to DNA. It will stain necrotic cells and cells in late apoptosis. It will not enter cells in early apoptosis nor living cells with intact membranes. So here's a little cartoon. As all of the target cells are initially labeled green with CFSE and the effector cells are not labeled, these two populations can easily be distinguished in a single tube. Again, you get live target cells and dead target cells. Keep in mind that control populations must be made to test the experimental condition and to compensate for the flow cytometer. So if your experiment is designed to determine cytotoxicity using isolated macrophages or monocytes or something, you also want to prepare a control of non-infected target cells combined with effector cells to determine what normal cell death would typically occur when interacting with healthy target cells. So in analyzing this, you would first derive a plot of the FL1 CFSE versus side scatter to identify all the green target cells from the unstained effector cells, which is number 1 on the left. Then you would create a plot of FL1, again, the green CFSE, versus FL3, which is the 7AD red, to further distinguish live green target cells on the bottom from necrotic red and green target cells on the top. You'd gate on all the green target cells in R1 and R2 and collect events on the right. Again, FL1 is for green, CFSE on the x-axis, and FL3 for red, 7AD, is on the y-axis. Live green target cells are in the lower right, which is R1, while dead membrane-compromised target cells are in the upper right, which is R2. Effector cells are on the left as they are unstained. Based on the percentage of green target cells in R1 plus R2, cytotoxicity can be calculated as the percentage of green target cells which are also stained red with 7AD in R2. This analysis reveals the population of necrotic target cells without interference from the necrotic effector cells, which are in the upper left. Traditional enzyme release assays are often skewed by that large number of necrotic effector cells, but this test really is not affected by those cells. ICT's basic kit is really the easiest way to assess cytotoxicity. In these experiments, K562 target cells, which are the human erythroleukemia cells, were stained with a CFSE and adjusted to 1.5 times 10 to the 4th cells per tube. They were exposed to gamma delta lymphocyte effector cells and added at different ratios and then incubated for just 4 hours. Cells were analyzed and plotted versus the effector to target cell ratio, and as you can see on the chart, cytolytic activity increased as more effector cells were added. To use ICT's total cytotoxicity in Apoptosis kit, just as with the basic kit, you would first start by labeling the target cells green with CFSE. And again, if studying the effects of a drug or toxic agent, the CFSE staining is optional if you're not going to be mixing it with the effector cells. Assuming you're mixing it with effector cells, you would add those next, which are unstained, and then incubate. After incubation, you'd add our SR-FLICA reagent. So SR-FLICA is a caspase targeting inhibitor peptide sequence of valine alanine aspartate, or VAD, which is flanked by sulforhodamine B and a fluoromethyl ketone group. The VAD sequence is a polycaspase amino acid sequence with affinity for most caspases. Caspases are an early and direct indicator of apoptosis. SR-FLICA will enter all the cells and bind to active caspases, thereby labeling apoptotic cells kind of an orangey red. If the cell does not have active caspases, SR-FLICA will simply wash out of the cell. It is not actually cleaved by the caspase. Instead, it will form a covalent bond with the caspase reactive sites, which renders that particular caspase enzyme inactive, and it can no longer cleave any substrates. Bound SR-FLICA will remain inside the apoptotic cells, and unlike antibodies, SR-FLICA will not react with procaspases nor inactive forms of the enzymes. Finally, add 7AD to stain necrotic cells red. So yeah, I wanted to show you a little cartoon picture of the 4 different populations of cells that you'll get with this. The target cells are initially labeled green with CFSE, and the effector cells are not labeled, so those are here in blue. The cells can be easily distinguished, and then by gating on just the green target cells, the flow cytometer can then separate the orangey red apoptotic cells from the red necrotic cells to identify early and late apoptotic target cells. And keep in mind that several control populations must be made to test the experimental conditions and compensate the flow cytometer. So when doing your analysis, you would first derive a plot of FL1, which is CFSE, green, versus the side scatter to identify the green target cells. Then you would create a plot of FL1, CFSE, versus FL3, which is 7AD, to further distinguish the green target cells from the unstained effector cells and the red cells. And then identify all the green target cells and gate on them as R3, which is R1 plus R2, and set an acquisition to collect your events within R3 to separate the orangey red apoptotic target cells from the red necrotic target cells, which is the 2 quadrants on the right-hand side, number 3. Here we'll look at this a little closer. Staining with SR-FLICA is really essential to quantify cell death, as 7AD simply will not detect cells in early stages of apoptosis. When used together, this test will reveal 4 populations of target cells. These are the live cells in the lower left, which are just stained green, the early apoptotic cells in the lower right, which are stained green because they're target cells, and orangey red with the SR-FLICA. And then cells in the late apoptosis, which are in the upper right, those cells are green because they're target cells. They're also red because they're necrotic, and they're a little orangey red because they have active caspases. And the necrotic target cells, which are in the upper left, those cells are just green because they're target cells, and they're also stained red with 7A-AD. A significant percentage of cells that are 7A-AD negative, indicating that they're live and do not have compromised membranes, but are SR-FLCA positive, which is in the lower right, meaning that they're becoming apoptotic and dying, they have active caspase enzymes, even though their membranes are still intact. So they wouldn't show up as necrotic. So based on the percentage of cells in each quadrant, total cytotoxicity can be accurately calculated. In this case, it's 32.59%. This analysis reveals that population in the lower right quadrant of early apoptotic cells that is not detected by any other method. 7.98% are SR-FLCA positive, but 7A-AD negative. These early apoptotic, soon-to-be necrotic cells can then be included in the overall percentage of total cell death, leading to more accurate results. By including all apoptotic events to analyze cytotoxicity, here which is the 7.98 in the lower right, 22.63 in the upper right, and 1.98 in the upper left, cytotoxicity can now be accurately quantitated as 32.59%. Other assays would calculate cytotoxicity only at about 24% because they would miss that lower 7.9% of cells that are in early apoptosis in the lower right, thereby underestimating the true level of cytotoxicity by about 25% of your total dying cells. ICT's total cytotoxicity and apoptosis detection kit is really the most accurate method to quantify cytotoxicity because it includes those cells in early apoptosis. Again, looking at the different ratios of effector to target cells, apoptosis occurred at all ratios. And ICT's kit detected the apoptotic cells, that's the blue line, leading to a more accurate assessment. Drugs and vaccines often work by stimulating the body's immune system. Conversely, opiates are known to have suppressive effects on cell-mediated immune response. Microglia are the macrophages of the central nervous system, the CNS, and are the primary target cells for mycobacterium infection in the CNS. In this paper, the authors used ICT's cytotoxicity assay to determine whether this opiate drug, morphine, alters the capability of the microglia in the CNS to activate gamma delta T lymphocytes. In this case, pigs were either vaccinated with BCG alone, which is live tuberculosis vaccine developed from Mycobacterium bovis, which causes tuberculosis in cows, or morphine plus the BCG. The data demonstrate that antigen-specific cytotoxicity was markedly impaired due to morphine treatment, which is the yellow bar on the right. In another recent publication using ICT's total cytotoxicity kit, researchers examined the ability of NK cells to kill target cells in the inflamed liver. NK cells were cultured in different media to stimulate conditions in visceral adipose tissue and inflamed liver. M199 is the control media. NK cells were added to K562 target cells at a ratio of 50 to 1. And this data suggests that NK cell cytotoxicity against K562 cells is unaffected by secreted factors in adipose conditioned media and liver conditioned media, whereas others have reported that cytotoxicity of intrahepatic natural killer cells is impaired in the inflamed liver of patients with chronic hepatitis C. Their data today suggests that NK cell cytotoxicity against K562 cells is unaffected by the secreted factors in this conditioned media, in the adipose conditioned media and liver conditioned media. And other data indicate significant alterations in the natural cytotoxicity receptor expression in the liver. This kind of shows that hepatic tissue microenvironments can really influence their natural killer cell activation. So some of the questions we explored today were, do drugs or viruses or vaccines or other treatments trigger a cell-mediated immune response? And yes, they do, and some of the diseases and viruses can really fool the immune system. And how do you measure that? You would use a battery of reagents and be sure to include a marker for apoptosis, such as using one of ICT's cytotoxicity kits to give you a more complete data about apoptosis and necrosis. In summary, assays are needed to understand the mechanisms of cell-mediated killing to help improve medical treatments. Traditional cytotoxicity assays do not provide enough data and often underreport the amount of cell death. ICT cytotoxicity kits overcome these issues, and our total cytotoxicity and apoptosis kit also incorporates a Cas-based protease marker to concurrently assess apoptosis to measure 4 populations of cells using the flow cytometry, including those cells in early apoptosis. Therefore, ICT's kits generate more reliable, accurate, and reproducible data. So in addition to these cytotoxicity products and kits, ICT has several online resources where you can learn more about the products and sign up for future webinars. We also have all of our manuals and some technical help online. Our technical support people are absolutely brilliant, and they would really love to talk to you about any of your experiments. Of course, again, you can visit us at immunochemistry.com or send us an email to help@immunochemistry.com, and you can call us at 1-800-829-3194. I want to thank everybody for listening to today's presentation. I hope this was helpful to you. We're also always interested in knowing what other additional topics you'd like to hear about. You can contact our marketing director, Jackie, for that. And as a special thank you for everyone who watched this webinar, you can get 25% off one of ICT's cytotoxicity kits, which will be valid through the end of October. If you haven't tried it yet, it's really a good opportunity to save some of your R&D budget and generate some great data. Jackie, do we have any questions out there?
Thanks a lot, Sally. Again, everyone, feel free to chat in your questions. We've got some limited time left, so I'll be fielding a few of the questions to Sally. But if we don't have time to get to your question, or your question requires a longer and more in-depth technical response, we will be following up with everybody after the webinar who chats questions in. So with that said, I'll go ahead and get started here. So it looks like the first question asked here, someone's wondering how they determine the percentage of cell death from their treatment using the cytotoxicity kits.
Oh, okay. I put those calculations up here. Let me go through that quick. In the basic kit, you can just create a gate around, let me find it here. You could create a gate around all the cells which are stained green, those are the target cells, and then count how many of them are simply stained red with 7A-AD, which is in the upper right. That will tell you how many cells are membrane compromised and dead. With the total kit, this really goes one step further by also counting the number of cells that are orangey red, as those cells are apoptotic. So again, you count the cells in the lower left, the lower right, and the upper right. Oh, I'm sorry, you count them in quadrant 2, 3, and 4 to determine which ones are necrotic and apoptotic.
Great, thank you, Sally. Someone else is wondering what type of controls are necessary when running their assay?
Okay, that was another slide I went through pretty quickly. Let me find that here. So with the basic kit, you need controls for your experimental cells, which would be treated and untreated cells. And then you also need some unstained and stained target cells so that you can gate on the CFSE positive cells, which are the B and C. And then some unstained live and dead cells, which are stained red as controls for the 7AAD. With the total kit, you also need to include some apoptosis controls, which would be non-induced and induced cells, and gate on those orangey red cells. Both of the kit manuals thoroughly really explain these controls, and if you have any issues, please give us a call. We'd really love to hear about your assay.
Thank you. Someone else is wondering a general question about apoptosis, but why not use an X5 to label apoptotic cells instead of using a Cas-based inhibitor?
Oh, okay. Well, looking at our data here from the total kit, ICT actually considered that, but an X5 can be rather nonspecific. There are certain features of its binding with phosphatidylserine which tend to lead to some false positives. The phosphatidylserine molecules are really inverted within the lipid bilayer of cells, for some reasons other than apoptosis. You may see many non-apoptotic cells become positive for an X5, even though they're actually not apoptotic. It's just a normal feature of some cell membranes, and caspases are really an early and direct indicator of apoptosis, while phosphatidylserine is a side effect and often appears late in apoptosis. Caspases are a much better indicator of apoptosis, and this is a case where you'd really want to consider, what's the artifact that you're measuring.
Great, and I think we have time for about one more question here. So the last question we'll take today, again, feel free to keep chatting them in if you still have some, but someone's wondering whether they can use these kits with fixed cells.
Well, you really can't use the fixed cells for the staining process, before you're doing your analysis. The kits are used with whole living cells. So you stain your cells with the whole living cells. Fixed cells won't work for the staining process because they're porous, which makes the reagents leak in and out. But you can fix them after you've stained them. The CFSE and SR-FLICA can be fixed, assuming that you've done some wash steps prior to fixation. 7-AD is the only dye that really would present a potential problem because it binds to the DNA of cells that have compromised membranes, and they might go in and out. It could still be compatible with fixation as long as you did the 7-AD staining before fixing, and any free 7-AD present in the buffer or cell media would have to be washed away. Otherwise, you might have permeabilized membranes, and you might accidentally get some that would otherwise have been excluded. So once the reactions have been completed and the wash steps have been done to remove any excess 7-AD, the cells can be fixed. Of course, your fixation itself has to be compatible or not interfere with whatever cell-mediated cytotoxicity process you're looking at.
Great. Well, again, thank you, everybody. That's about all the time we have for today. Again, if we're unable to get to your question, we'll follow up with you personally right after the webinar here. Again, I'll be sending out the webinar recording within the next day or so. So if you missed anything today or would like to refer back to the slides, you'll get that within an email from me. Our next webinar is scheduled for October 26th and will be on the topic of cathepsin detection in real time. And you can register for this webinar right now. That registration link is live on our webinars page. If you can think of any other questions after the webinar or have ideas for future webinars, please go ahead, fill out the Contact Us form on our website, or you can reach us through social media on Facebook, LinkedIn, or Twitter. We're really happy to get your feedback. Thank you again for joining us, and I hope everyone has a